Welcome to another class. So in this class, we'll be looking at question 3. So question 3 is talking about blood and blood transfusion. So you need to go through this topic. So this topic, for those of you that don't know where it is under transport in human beings. That's where you find the topic of blood, and it has also the same table, so make sure you go through the table. So the question is saying the table shows two blood group A, B and O and their properties. The first question is complete the table by filling in the spaces provided. So how can you fill in the spaces provided? So imagine that you don't know anything about blood. How will you know what antigens are? How will you know what antibodies are? So before we begin solving the table, I will explain to you what it means by antigen and what it means by antibodies. So when something is an antigen, is basically a foreign substance to your body, usually on the surface of cells. So this antigen attaches itself to surface of cell. Imagine this is your cell, then an antigen comes and it attaches there. So once an antigen attaches to your cell, it will stimulate the body's immune system to produce what are known as antibodies. So which brings us to our second definition. What is an antibody? So an antibody is just a specific protein which will be produced now to destroy that antigen by binding to it and neutralizing it. So what have we talked about? So we say that the first thing is an antigen is a foreign substance. Then an antibody is what? Is the response that the body will give this foreign material so that it destroys it. So by this definition, I guess we have now understood what it meant by antigens and antibodies. So now imagine if you have a blood group of AB. What do you think are the antigen? So if you have a blood group of AB, of course it should have antigen A and antigen B, because we have A and B. We can't have A and B, then we have antigens like O or other antigen. So meaning blood group A has antigen A and B. So these are the two antigens which are present in what? In blood group A, B. So what do we mean? So what do we mean is that, for example, if someone has what? Antigen A, then you put that blood in someone with blood group B, meaning that A will be attacked because it will be identified as a foreign material in someone who has blood group B. So remember that. Then antigen A. So antigens just follow the blood group. So if it's A, it has antigen A. If it's B, it has what? Antigen B. Then if it's AB, it has both. But blood group O has no antigen. So blood group O has no foreign substance. So meaning blood group O can be transfused into anyone. That's the reason you get blood group O to be the universal donor. Yeah. Then moving on to our antibodies. So what are the antibodies? So we say that these antibodies are the ones now which are produced against what? Antigens. So now what are the antibodies? So meaning if blood group AB has antigens A and B, meaning it will not have antibodies, because someone who has AB has both what? Antigen A and antigen B. So there will be no need of us attacking what? The blood group. So meaning someone with blood group AB has no antibodies. So here you can say none. Then when it comes to blood group O, so blood group O contains what? Antigen A and B. So we have anti A and B. Yeah. So what do we mean when we say blood group O has antigen or have antibodies A and antibody B? So someone who has blood group O cannot receive what? Blood from A or B. Why? So since this person does not have any antigens which will help his body to recognize that, oh, A is not a foreign material, B is not a foreign material, it will start to attack A and B because they are not present in what? In the person's body. So meaning that's why you can not give someone blood. So you cannot donate, or that's why blood group O only receives blood from O because both don't have antigens. So that's how you can fill out the table. So next question is saying, explain what an antigen is. So we are from explaining what an antigen is. So what did we say an antigen is? We said that this is a substance or you can say a foreign material on the surface of cells which stimulates the production of antibodies. So that is the definition of an antigen. Is just basically a foreign material, something which has entered your body that is foreign. So something foreign is something which is not part of your body. Then part B is saying, explain why a person with blood group AB can receive blood from any donor. So we said that the reason is that blood group AB has no antibodies in their plasma. Therefore, the A and B antigen on the donated blood cells will not be attacked. So if someone has no antibody, meaning if they get an antigen A and antigen B, they won't be attacked. So you can say blood group A has no antibodies in their plasma. Therefore, the A and B antigen won't be attacked. Yeah, as a result, it will receive blood from A and B. Part C. Determine the effect of blood from a donor with blood group AB being transfused into a recipient of blood group O. So now here, they're asking you, when they transfuse blood now, AB, to someone with group O, so the recipient and A and antibody will react now here. So we said that O has both the antigen. So you can say since O has both antibodies for A and B, it will cause what? Agglutination. So what is agglutination? So agglutination is just the clumping of blood. And also we have hemolysis, which is the destruction of red blood cells. So this is what can happen when you donate blood group A to someone with blood group O. So those blood will start to what? To clump. We start to joining. We start joining together, forming clumps, and the antibodies will start attacking the what? The blood in the donor's blood. So therefore, it will cause now the hemolysis and clumping in the recipient's blood. Therefore, he can die. So the last question, which says, explain the importance of screening blood for the purpose of transfusion. So the first thing is the one which you are from saying. So you have to test for blood what? Compatibility. Of the blood group, so that we prevent what? To prevent agglutination and clumping. Then the second thing is to detect infectious disease. So for you to detect infectious disease, you have to screen the blood. So these are the two types, these are the two importance of for you to screen blood before you give someone. So this is where our question 3 ends. Don't forget to subscribe and comment if the video was